Hello, Akshaya.
Good evening, ma'am. Good evening,
Good
sir.
evening, ma.
Good evening, Ajay.
Yeah. Today we are going to do the first unit. We are going to just look at some summary of the first unit, first chapter of the first unit in 12th standard CBSE book. அதாவது reproductionல வந்து first we are going to see sexual reproduction in flowering plants, angiosperm. And then we will see a little bit of human reproduction also. I don't know if if we will have time to finish the entire unit today. So we will do a little bit half the unit today and rest of the unit in the next week. Then after that we can consolidate everything and do neat questions. Discuss the neat questions in this unit. Okay, so we'll right away go to the chapter. Are you all able to see this?
Yes, madam.
Yes, ma'am.
Okay. Now, isn't it? Sexual reproduction in flowering plants, angiosperms, and the specialty of the angiosperms, they produce flowers, seeds, and so most of the plants, I mean, almost all the flowering plants, they show sexual reproduction. So actually, the site where the sexual reproduction actually happens is the flower. So we may think the flowers are for others to see and it's so colorful and it's for the benefit of others. No, actually it's for them. It's actually they are using it for the reproduction. Okay. Now, what is the structure of the floral parts actually? The floral, I mean, when there is before the actual flower matures and it's produced, in the floral primordium, it has, I mean, it sometimes it's like a inflorescence bearing floral buds, and then from that flowers come. And in the process, some hormonal and structural changes result in differentiation and development happens. So we will see all that now. So actually, I mean, it starts with a small bud and then it enlarges and becomes a flower and get matured, and it then gets ready for the pollination. Now, when when you take the flower, you should know. I mean, there are so many things we know about this. I mean, the androecium, gynoecium. Androecium is the male sexual organ of the flower. Gynoecium is the female sexual organ of the flower, and how they develop into male gamete and female gamete, and then how they fertilize, and then the pollination happens, and then the fruit or seed is developed, and from that how it germinates. So this is all about the sexual reproduction of the flowering plant. Now, when you take a typical flower, it has two parts. I mean, there are many types. I mean, some flowers are unisexual, some flower are bisexual. And I mean, there are different types. And then as and when we see or read more about this, we will discuss about that also. Now, a typical flower means majority of the flowers has two parts, androecium and gynoecium. Now, androecium is a male sexual or male reproductive part. Now, what does it consist of? It consists of a stamen or whorl of stamens. Well, okay, so their length, number and length are variable in different species. I mean, some may have one or some may have more, many. I mean, it depends on the species. Now, this stamen is the what to say? It consists of filament and anther. Now, in this anther, see the parts of what are the parts of the stamen? It has filament and anther. Filament is this long stick-like thing. You can see the here. It's this part. Are you able to see that my cursor? I'm showing it with the cursor. This green color stick-like thing is the filament, and the tip is the anther. Usually, this anther carries the pollen grains. I mean, when matured, but before maturation, it will have microsporangium. This anther most probably will be bilobed. Can you see? This is bilobed. It's like this: two lobes, bilobed, two lobes, and the filament's proximal end is attached to the thalamus or the petal of the flower, and the other end is the anther, which is bilobed. This each lobe has two thecae, that is dithecous. So often the longitudinal groove runs lengthwise, separating the theca. Here, it goes lengthwise, separating the theca. And now here, you have the microsporangium, which develops into pollen grains. You can see the cross section of that. So here, these are the pollen sacs or the microsporangia. And the microsporangia has the microspore mother cells. Now, this pollen sac or the microsporangia is enclosed by three layers. Those three layers are epidermis, endothecium, middle layer, and tapetum. See, the outermost is the epidermis, and then next is the endothecium. I mean, this will be clear here. Yeah, it will be clear here. Is it clear? Now, epidermis, then endothecium, then middle layer, then the light green color is the tapetum, and then inside you see a lot of dots. I mean, just for the illustration, he has given like that. It's not going to be like that. But just for you to understand, he has given the diagram like that. So that is the microspore mother cell. It's packed with microspore mother cells. I mean, like this, there are four. So each lobe has two. That's why ditheca. All right. Now, what will be the structure of the microsporangium? The tapetum or the innermost layer gives a nourishment to the pollen grains. Now, when the pollen grains are formed, it gives the tapetum gives the nourishment to the pollen grains. So the cells of the tapetum contains dense cytoplasm and generally have more than one nucleus. Okay, like this. This is the tapetum layer. You can see at the bottom in the diagram, this is the tapetum. It is dense. I mean, there is no gap. You can see that it's dense with cytoplasm and it has more than one nucleus. In the young anther, that is before maturation in the anther, each microsporangium, that is even, I mean, you can see four, isn't it? Each one we are talking about. There are four and one we are talking about. As porogenous tissue at the center. So at the center, it has porogenous tissue. and it consists of compactly arranged homogeneous diploid cells, that is sporogenous cells. And as the anther develops, what happens is each sporogenous cell, that is a microspore or the pollen mother cell, undergoes meiotic division to form microspore tetrads, like here. You can see this will be a tetrad. You can see only three, but in the back side. So it will be like a ball. Four haploid cells are formed. Because in meiotic, haploid are found, isn't it? So the microspores arranged in a cluster of four cells. So that we say tetrad. The formation of the microspore from a pollen mother cell, that is, we call it, they generally refer to as PMC. So when in question or anywhere, if you are referred to PMC, then you should understand it's pollen mother cell. Through meiosis is called microsporogenesis. Okay, so from the anther, each sporogenous cell or the microspore pollen mother cells undergoes meiotic division to form microspore tetrads, and then the formation of microspores through the meiosis, that process is called the microsporogenesis. Okay, all right. Now, microsporogenesis, what is it? Um, like as See, as the anthers mature and dehydrate, I mean, the more and more it gets matured, it get automatically it gets dehydrated, and then it starts dissociating with each other. I mean, the closed, compact arrangement goes, and then it starts dissociating. Only then it can be let loose, and it can go out to fertilize. So dissociate with each other and develop into pollen grains. So like here, you can see here in the diagram. So each microsporangium contains thousands of pollen grains. They are released with the dehiscence of anther. So from the anther, it is released. So about the pollen grain, how it is? No, each species or each plant, it depends on the plants. In some plant, I mean, they exhibit different shapes, sizes, color, and everything. And of course, it's microscopic. So this is generally spherical. Generally, it's spherical because it will be easy to, easy to be transported, and it's 25 to 50 millimeter in diameter, and cytoplasm is surrounded by a plasma membrane. A pollen grain has two layers called exine and intine. This exine actually has few openings. It's covered with a very thick, and I mean it cannot be destroyed. It's made up of a very organic material, high resistant organic material called sporopollenin, and it can withstand very high temperature and strong acid and alkali. So enzyme cannot degrade sporopollenin. That is the exine, that is the outer layer of the pollen grain. Now this has some apertures, that means openings, at several places in the pollen grain. Those apertures are called gem pores. So actually, when the pollen grain is transported to the stigma, from the pollen grain, pollen tubes are formed through this germ pores or the apertures. Okay. So the pollen grains are preserved as fossils due to the presence of sporopollenin. Because I mean, it cannot be. It's an organic material, very highly resistant organic material. It cannot be degraded. So it's preserved like a fossil. So exine exhibit pattern and design. So this exine only exhibits the different patterns and designs and colors of the pollen grain depending on the species. Now this pollen grain is the male gametophyte. Now in the interior, the wall will be intine. That is a smooth, thin, continuous wall. I mean, the layer made up of cellulose and pectin. So, any matured, when it develops into a mature pollen grain, which is ready for fertilization, contains two cells, sometimes three. So, this two, anyway, we will come to it, why it's three. Now, this has two cells. One is vegetative cell and another one is generative cell. The vegetative cell is a big one with abundant food reserve and a nucleus which is irregular in shape. The generative cell, actually, this vegetative cell supports the generative cell. It gives it nourishment actually, and this generative cell, it's spindle shaped with dense cytoplasm and a nucleus. That's all, and it is small and floats in the cytoplasm of the vegetative cell. So it consists of two cells within the pollen grain. And over 60% of the angiosperms shed their pollen grains at two-celled stage. The remaining of them, the pollen grains are shed at three-celled stage. Some of the plants. Okay. The shed pollen grains have to land on the stigma before they lose viability. So once they are out, their viability period is very limited. Some have very long viability, but some have very short viability. Like, for example, the viability of the pollen grains of some cereals, rice, wheat, etc., it's only 30 minutes. So within 30 minutes, they have to go on, land on some stigma of some other flower. And this viability also depends on temperature and humidity of the place. Some members, Leguminaceae, Rosaceae, Solanaceae, have viability for months. Okay, all right. What is the economic importance of pollen grains? Pollen grains has, as we saw, it's very nutritive because it has a lot of food stored for the generative cell to support the generative cell. So it is rich in nutrients because that has to grow, isn't it? That's only is going to facilitate fertilization. So it's a very important to nourish the generative cell inside the pollen grain. So it's rich in nutrients. And people, actually, it's frozen and then it's used for tablets and for food, in food supplements. As food supplements, some of the food supplements they use. Pollen tablets and syrups increase performance of athletes and race horses. And they are stored for years in liquid nitrogen, that is minus 196 degrees Celsius. They are used as pollen banks for crop breeding programs. Some of the pollen grains, like for example for Parthenium or carrot grass, they find it very allergic. Some people are allergic. Like for example, you see in Bangalore, you have a lot of Parthenium and carrot grass, and many people get allergic to that, the pollens. It's just one place I'm mentioning. There are many places where these pollen grains become allergic. When this particular species of plants are in abundant, I'm saying. Okay, so what is the effect of the allergy? It can lead to chronic respiratory disorders, asthma, bronchitis, etc. Now, the female reproductive part, the gynoecium of the flower, that has it's called pistil. So this pistil consists of style, stigma, and ovary. So the ovary is attached to the thalamus. Now stigma is you can see In this diagram, the first one is hibiscus pistil. This is monocapillary. And this one, if you see, this is multicapillary syncarpus pistil of papaver. The third one, C, is a multicapillary apocarpus gynoecium of Michaelia. So, when you say multicapillary syncarpus, the pistils, I mean, there may be more than one pistil. You can see hibiscus has only one pistil. You might have seen the hibiscus flower. It has only one pistil. But there may be flowers which has more than one pistil. So, and it can be syncarpus or apocarpus. Apocarpus means syncarpus is all the pistils are attached, fused together. Apocarpus means they are all free. I mean, they are individual. So this each pistil has three parts, as I told you. This ovary, that is at the base, bulged part, and style is the elongated, slender part, and stigma is the landing platform for pollen grain. Is the top part here. This part. Okay. So inside the ovary is a ovarian cavity, or that cavity is called the locule. In the locule, it has placenta, and actually this placenta, the ovule inside the ovary is attached to the placenta. So arising from the placenta are the ovules, or these ovules are the megasporangia. When you see androecium, it's microsporangia, and here it is megasporangia. The number of ovules in an ovary may be one, as in the case of wheat or paddy or mango, or many, as in the case of papaya, watermelon, or orchids, etc. Now, if you see the megasporangium or the ovule is attached to the placenta by stalk, that is the funicle. So this is the funicle. Are you able to see the diagram? This is the funicle and this is the placenta. And in this placenta, it has the ovary or the embryo ovule is attached, and this has two protective envelope or the integuments, outer integument and inner integument. And except at the tip where a small opening, that is micropyle is present. See here, this is a micropyle here. Micropyle is present. This is the micropylar pole, this place where the opening is there. Opposite to the micropylar end is a chalaza or the base here, this side, opposite side. This is a micropylar pole and this is a chalaza or the basal part. And you can see here the ovary wall, placenta is this one, and the ovule is attached to the placenta through the funiculus or the funicle. Now the structure of megasporangium, see, we will look at it more closer. Now, this is enclosed within the integuments that we saw, outer integument and inner integument. Two layers are there. And there is a mass of cells called nucellus here. This one, this part. After the inner integument, there's a mass of, place where the food reserve is there for the development of the female gamete. So that is called the nucellus. And the center part is the embryo sac. An ovule generally has a single embryo sac formed from a megaspore. Now here, see, it is a formation of, actually, what is megasporogenesis? Megasporogenesis is the formation of megaspores from the megaspore mother cell. Okay, So the ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and prominent nucleus. Here, this one. Okay, this is a large cell containing dense cytoplasm and a big prominent nucleus. So the megaspore mother cell undergoes meiosis to produce 4 megaspores like this. Can you see? And this in turn again undergoes meiosis to produce totally 8 megaspores. Okay, all right. This we call megaspore tetrad, as in the case of pollen tetrad. Okay. In majority of flowering plants, one megaspore is functional while the other 3 degenerate. The functional megaspores develop into female gametophyte. So the embryo sac formation form a single megaspore is called monosporic development. Here, this tetrad, only one will be functional, the other 3 degenerate. And the embryo sac formation from a single megaspore is called the monosporic development. And this nucleus of the functional megaspore divides mitotically to form 2 nucleus. They move in opposite poles, forming 2 nucleate embryo sac, like as in here. And then the nuclei again divide 2 times, forming 4 nucleate and 8 nucleate stage of the embryo sac. So from this to here, and then again it divides and becomes 8 nucleate. These divisions are free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. So what happens is, and when the cell wall forms, I mean this here after 8 nucleates, then the cell walls are laid down, leading to the organization of the typical female gametophyte. 6 of the 8 nuclei, what happens is, are surrounded by the cell wall and organized into cells. The remaining 2 nuclei stay in the center. That is, those are the polar nuclei. They just stay in the central cell, below the egg apparatus. So this will be the egg. Below the egg apparatus, it will stay in the central cell. So the near the egg apparatus, the 2 nuclei is the synergids, and then the 3 on the other opposite side are the antipodals. And there is a filiform apparatus is there, which will guide the pollen tube, or the male gametophyte into the synergids. So that is the function of the filiform apparatus. So now, a typical mature embryo sac is 8 nucleate and 7 celled. So how many cells are there? 3, here 2, and then 1 egg apparatus, 5, 6, and then on the central cell 7. But totally how many nucleus are there? 8, because there are 2 polar nuclei in the central cell. So 3 cells are grouped at the micropylar end and form egg apparatus. It consists of 2 synergids and 1 egg cell. The synergids have special cellular thickening at the micropylar tip, called the filiform apparatus here. So, and then the 3 cells at the chalazal end are called the antipodal. The large one in the central part is a central cell that has a polar nuclei. All right, so this is about the female embryo sacchinicium. Okay, now the formation of female gametophyte. Overall, what happens? So this first, by meiotic tetrad is formed, and then of the four, three degenerates and only one becomes functional, and then that the nuclei divides and becomes eight nuclei, and in that three two stays in the middle, in the central part, to be the polar nuclei, and then three becomes antipodal, and then two synergids and one egg cell. Okay. Now with this pollination. So pollination, you see, there are different types of pollination. So basically, it's transforming the pollen grains from the anther to the stigma of the pistil is the pollination. That you all know. So there are three types of pollination. First is autogamy. What is autogamy? That is self-pollination. In the same flower, from anther to stigma. This, pollinating the pollen grains to the stigma. That is the transforming. This is the A1. In the same flower, from the anther to the stigma of the same flower is a autogamy or self-pollination. Now, in the same plant, from anther of one flower to the stigma of another flower is a detinogamy. And then anther of a flower of one plant to the stigma of the flower of the same species of the another plant, that is a xenogamy. So these are three types of fertilization, pollination. At this point, I have to tell you this chasmogamous flowers and cleistogamous flowers. So cleistogamous flowers are something which don't open, and completely it don't open at all. So it actually fertilizes inside itself. Within the same flower itself. Whereas the chasmogamous, it's not self pollination. It's not actually self pollination. Both may be in the same plant. Both type of flowers may be in the same plant. Okay. But repeatedly, this autogamy, may depress the inbreeding or depress the, species, newly formed some, germination, maybe depress, or the plant or whatever species is produced may not be very healthy. It may depress the breeding. Gytonogamy is, it is as we saw in the same plant from different flowers. Okay, so that is it's not actually in straight away you can't say it's autogamy, but genetically yes, it is autogamy kind of thing. Then because it belongs, both the flowers belong to the same plant. But sometimes when so how this happens? See, this fertilization, pollination, actually they are dependent on external pollinators. It can be abiotic agents or biotic agents. The pollinators, who facilitate pollination are the agents or the pollinators. It may be abiotic or biotic. Abiotic is what? Wind and water. Sometimes like lower plants like bryophytes or lower plants may depend or may require water for the for the male gamete to float to the female gamete for pollination. And even some other plants other than the lower plants, they use water as a medium for or agents for pollination. And some plants they use wind. So pollination by wind is anemophily, more common abiotic agent. So this, like for example, if you see corn cob, the tassels are the stigma and style which wave in the wind to trap the pollen grains. So the wind pollination is quite common in grasses. So ways of effective pollination is the flowers produce large amount of pollen because I mean like it may be wasted many, so it has to produce more. So the pollen grains are light and non-sticky. I mean when the wind blows it should be able to be carried away by the wind very easily. So they often possess well exposed stamens for easy dispersion of pollens into wind currents and large feathery stigma to trap airborne pollen grains. That's all these things. They are um, um, they're prepared for this. I mean, the plants which use wind as a agent. They're
கை பிடிச்சிட்டு கை பிடிச்சிட்டு தெரியாதா இப்படியெல்லாம் இருக்கக்கூடாதுண்ணா
என்னையா பேசுறேன் அப்பா சொல்லுங்க நீ எனக்கு தினம் எனக்குன்னு அவங்களாதான் எனக்கு கூப்பிட்டாங்க நான் சரி அத்தை கீழே கீழே முந்துருவான்னு அப்பதான் சொல்லிட்டு இருக்கிறேன் அதுங்களே எப்படி கீழே விழுந்துருச்சு
yes, Selvaraj. All right. So the one thing about this is the flowers are not very colorful. They don't need to, produce nectar. I mean, for abiotic agents, wind and water, they don't need to produce nectar, or I mean, some incentive when you depend on the biotic agents like insects or animals to transport the pollen grains, you have to give them some incentive like nectar and beautiful colors for attracting them. All these are necessary, but this wind and water agents, it's not necessary. So such plants which depend on the abiotic agents may not have all these things like attractive color or nectar or anything. Okay, pollination by water is hydrophily. So some plants, I mean, are limited about 30 genera, mostly monocotyledons. They are quite rare. They depend on water for pollination. Vallisneria, the female flower reaches the surface of the water by the long stalk, and the male flowers and pollen grains are released onto the surface of the water. They float and then they pollinate. And then the sea grass, actually the female flower remains submerged in water and the pollen grains are long and ribbon-like here. Ribbon-like, and then they are carried inside the water and reach the stigma. So that's how the water pollination happens. For I mean, these are for lower plants only. Mostly for lower plants, even biophytes, they need water. And then biotic agents, animals use majority of flowering plants. I mean, they are very, very colorful. What are the agents? I mean, who are the agents for this? Bees, butterflies, flies, beetle, and so on. So many are there. So even indirectly, even humans, because we take the fruit and then eat the fruit and throw the seeds. That way, we are facilitating germination. But we may not be helping them to do the pollination. So pollination by insects is entomophily. Particularly bees is more common. So when they depend on the entomophily, they have to give them some incentive like nectar, which will bring them to the flower and then it will pollinate them automatically because the pollen grains are very sticky. Once they sit on the flower, it sticks, and then when it goes to the next plant, and then automatically it's fertilized. I mean, the pollen grain is transported to the stigma of the another flower. Okay. See, the incentive may be different. Not only nectars, some flowers may be very big to accommodate, for the insects to lay eggs. Like, for example, a moth species and the plant yucca. This plant has the tallest flower for 6 feet. Amorphophallus. And another moth species and the plant yucca cannot complete their life cycle without each other because moth deposits its eggs in the locule of the ovary and the flower gets pollinated by moth. The larva comes out of the egg as seeds start developing. Actually, sometimes the insects consume the pollen or the nectar without bringing about pollination. So such insects, when they say it's pollen or nectar robbers. All right. Now, this hermaphrodite flowers can undergo self-pollination or the autogamy. Continued self-pollination results in inbreeding depression that we saw. So to avoid self-pollination and encourage cross-breeding, there are some devices in plants. Now, what happens is avoid synchronization. What they do automatically, even in them, they are built in like that. See, when the pollen grain gets matured, the stigma may not be ready. I mean, stigma of the same flower may not be ready yet for the pollination. So, I mean, or when the stigma may be ready, but the pollen grains may not have matured. So, the timing is different. That's what it means. It's not in sync. So, the arrangement of anthers and pollen different positions, they may be situated at different position, like you can see in this flower. It's not very close. It's far away, or one may be taller and one may be shorter, something like that. And then self incompatibility, like when a pollen falls on the stigma, if it's not compatible, it will not allow the pollen tube growth inside the stigma. So, or the germination, it will not allow the pollen germination. So, that is self incompatibility. So, that way it's preventing the self pollination. And the production of some plants like papaya, castor, and maize, it produce flowers present in the same plant. I mean, male and I'm sorry, this is unisexual flowers. I mean, like male and female flowers are present in the same plant, like castor or maize or whatever. And some plants they produce dioecious. That means one plant may have male flowers and completely, and the other plant may have female flowers, like for example papaya. So I mean, only the one which has the female flowers will have more fruits. The male plant, the plant which has the male flowers may not grow more fruit. So this prevents autogamy and gycynogamy. Now the pollen pistil interaction, how it is done. So it is in the process with pistil recognize the compatible incompatible pollen. Through the chemical components produced by them, they recognize. So what happens is if the pollen is compatible, it's a right type. That means the right type, whichever, I mean, it can, grow or germinate on the stigma. So the pistil accepts it and promotes post-pollination events. So what are they? The pollen grain germinates on the stigma to produce a pollen tube. You can see here, the pollen tube goes into that, and then through the Philippi apparatus, it goes into the synergids into the egg cell. Okay, so when it goes to the synergids, the male gametes are released. Actually, the pollen is released at the two cell stage. Most of the 60% of the pollens are released in the two cell stage. Some are released in the three cell stage. So, what happens to those two cell stages? When the pollen germination happens, the third cell is it divides and then produces the third cell, and then as the pollen tube is formed, that is also the third cell is produced, and then it goes through the Philippi apparatus and then to the synergids, and before it goes to the egg cell, the male gametes is released. So one of them fuses with the egg cell, the other one fuses with the polar nuclei. See, the artificial hybridization is they design pollen grains are pollinated with the stigma, like to for better yield or better species, to develop a hybrid variety. So this is achieved by two techniques. I mean, emasculation and bagging. Emasculation is just taking out. I mean, if you want the stigma, if you want to keep the stigma, you just take out the anthers and then take the pollen grains from other plant and then dust it on the stigma of the one first plant. That is emasculation, just completely removing one of the either male or female reproductive part. They remove it, either stigma or the style. I mean, they will remove it. Another one is bagging. They just bag it. Emasculated flowers are covered with a suitable bag and then prevent from contamination, prevent that from again pollinating or anything. I mean, they will not have any choice. They have to fertilize. It's like that. And then the mature pollen grains or anthers are dusted on the stigma for fertilization. So this, I mean, when the pollen grains germinate on the stigma by developing pollen tube, and then it fuses one of the, I mean, in the process, I mean, two male gametes, one of them fuses with the egg cell and the other one fuses with the polar nuclei. So this type of, I mean, this you all know, this what to say, pollen grains are haploid cells. So the egg cell is also haploid because that also is divided. Is multiplied and got by meiotic. So that is also haploid. So what will happen is it's called the fusion of the male gamete. One of the male gamete with the egg cell is called the syngamy, to form the zygote. That forms a zygote. Zygote is it's two haploid cells. It forms a zygote. It becomes a diploid cell. Okay, and then the other male gamete fuses with the two polar nuclei located in the central cell. That becomes triploid, and because three haploid cells, it becomes triploid, and that becomes the primary endosperm nucleus. And the cell becomes a primary endosperm cell and involves fusion of three haploid nuclei, and therefore it is called triple fusion. Okay. Now, These are two types of fusions, that is syngamy and triple fusion. It takes place in one embryo sac. So that's why we say double fertilization, and this is a characteristic feature of the angiosperms. So it is, yeah, as I say, it is unique to the flowering plants. So the central cell where the triple fusion happens is called the primary endosperm cell, that is a PEC. Now this develops into endosperm and zygote develops into embryo. See, this zygote then develops into globular embryo, then heart-shaped embryo, and then into cotyledon. So it is a that become this stage is the last one, is a mature embryo. At that stage, it starts sprouting, germinating. Now see, the primary endosperm cell divides repeatedly to form a diploid endosperm tissue. Okay, and the endosperm cells are filled with reserve food material they used for nourishing the developing embryo. So, in common endosperm development, the pen undergoes successive nuclear divisions to give free nuclei. This stage is called free nuclear endosperm. Um, I mean, the number of free nuclei varies greatly. I mean, it depends on the species and the plant and the variety. The endosperm becomes cellular due to cell wall formation. So, if you take coconut, the kernel part is also endosperm, the liquid part is also endosperm. The liquid part is the free nuclei stage, and the kernel will be the cellular. The endosperm becomes cellular due to cell wall formation. So that's why it is so thick. So both are endosperms in coconut. So I think, yeah, dicotyledonous embryo. I mean, it has two cotyledons. So it has an embryonal axis and two cotyledons. This is the embryonal axis, and it has two cotyledons. Two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyl, which terminates with the plumule or the stem tip. Here, the cylindrical portion below the level of cotyledon is a hypocotyl. Here, this is a hypocotyl, and then that terminates to the radicle or the root tip. The root tip is covered by the root cap. So that's all about the two cotyledons. Monocotyledon is slightly different. They possess one cotyledon. Okay, here has one cotyledon, or it is called, scutellum. It is actually lateral to the embryonal axis, and then it has a radical. Root cap is enclosed by coleorhiza. This is the root cap. Coleorhiza is below the root cap, and then it has epicotyl, then shoot, apex, leaf, primordia. All this is there, and this space is hollow foliar structure. So mature seeds are of two types. So non-albuminous seeds, they have no residue endosperm as it is completely consumed during embryonic development. And albuminous seeds retain some endosperms like wheat, maize, barley, etc. Okay, those are two types. And then what else is there for you? Occasional in some seeds, black pepper, beech, etc. Remnants of nucellus are also persistent. That is called perisperm. So integuments of ovules harden as tough protective seed coats. It has some small pore through which water and oxygen enter into the seed during germination. So moisture is taken. I mean, once some seeds, they become very dry and dry and they reduce water content. And that is 10 to 15% moisture by mass. I mean, what happens is because of the metabolic activity of the embryo slows down. So it reduces the water content and then it goes to inactivity, dormancy stage. So what happens is such dormancy stage may be for many years. It can be there for many years. See here, some seeds can remain alive for hundreds of years. The oldest is that the lupine excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. So even for 10,000, this is the one. And then 2,000 years old viable seed of the date palm. So this is the date palm. Is discovered during the archaeological excavation at King Herod's palace near Dead Sea. So there may be seeds which go into dormancy and still retain the viability. All right. Now fruits from ovary. The ovary develops into fruit. That we all know. Transformation of ovule into seeds. The ovary into fruit proceeds simultaneously. So both happen simultaneously. Like for example, ovary into fruit and ovule into seeds happen simultaneously. So the wall of ovary develops into pericarp, wall of fruit. The fruits may be fleshy or dry. Fruits are of two types. True fruits or false fruits. What is true fruits? True fruits are the one which develop from ovary. And other floral parts degenerate or fall off. Most of the plants are like that. Some plants, the false fruit develops from the why? Because even the other part of the flowers also take part or contribute in fruit formation. That is, for example, thalamus, where even the thalamus may get involved in the fruit formation. Like for example, apple, strawberry, cashew, etc. The main fruit what we eat is the thalamus. And then there are some species of fruits developed without fertilization. Such fruits are called parthenocarpic fruits. Example, banana. This partheno can be induced through the application of growth hormones. Such fruits are seedless. Is not very good. That's why they say seedless fruits are not very good for consumption. It may induce some hormonal imbalance if consumed more. The apomixis is the production of seeds without fertilization. Some species, Asteraceae and grasses, it form asexual reproduction that mimics sexual reproduction. Actually, it happens, the embryo without the development of seeds, how it happens is it sometimes without reduction division, it develops into embryo without fertilization. So what happens is it may have more, in many species, many citrus or mango varieties, some nuclear cells surrounding the embryo sac divide and protrude into the embryo sac to form embryos. So, thus, each ovule contains many embryos. Such cases. See, you can see here one seed, but many germination, many germinations come, multiple embryos. Can you see? This is called polyembryony. You can see here, single seed, many embryos. So if the seed from hybrids are sown, the plants in the progeny will be segregated and lose hybrid characters. So production of hybrid seeds is costly. So hybrid seeds are also expensive because they pick out the best characters and then they pollinate, and then since all these things it's pretty costly. The hybrid seeds are also, that variety is also costly. If the hybrids are made into optimics, there is no segregation of characters in hybrid progeny. So the farmers can keep on using hybrid seeds to raise new crop. So with this, we end this chapter, sexual reproduction in flowering plants. Shall we move on to the next chapter, human reproduction? All of you?
Yes, ma'am.
Good. So was this summary useful to you all?
Yes, ma'am.
Okay. We are not going to do much in human reproduction because I don't know if we will have time if we start to finish. So we will just see the introduction to human reproduction, and then just to see the male reproductive organs. Okay, just that. Then rest of the unit we can discuss in the next week. All right, the human reproduction, it's a production, the reproduction is the production of young ones by the organisms, all that we know. So humans are, many organisms are viviparous, and humans are also one of them, sexually reproducing and viviparous. Now, you may be knowing what is viviparous. Viviparous is carrying the younger ones till they are mature. They have the fertilization happens inside, and then they carry the young ones till they are born. So human reproductive system consists of two systems, male reproductive system and female reproductive system, and we are going to see today only male reproductive system. This male reproductive system has two parts. Actually, one is the external genitalia and the accessory glands, and then another thing is the paired testis and accessory ducts. So these are the important parts of this reproductive system. Now, what does it contain? The genitalia and the accessory glands has the prostate gland, that is the first one, and seminal vesicle, and Cowper's bulbourethral gland. So prostate gland is one, and seminal vesicle 2 is there. Cowper's bulbourethral glands again 2 is there. And here, accessory ducts. What are all there in the accessory ducts? Reti testis, vasa efferentia, epididymis, and vas deferens. Now, if you see, now I want to show you a different diagram to explain this. One second, children. Are you able to see this one? Can you all see what I'm sharing? Now can you see? Are you all able to see, children? Are you all there?
Yes, ma'am.
Okay, are you all able to see what I'm sharing now?
Yes, ma'am.
Okay. See now, in this, what we have to see is the external genital with the scrotum. Scrotum has the testis, and then this external genital is connected to different ducts, and then inside the testis, it's like this. There are two testes are there, and then inside when you cross section, it has testicular lobules are there here, and then from that comes the reti testis, and then that is taken outside through the vasa efferentia. It's a small gland, tube. It's taken to the epididymis here, and then that is connected to the vasa deferens. This is again a gland. Okay. Now this goes to the seminal vesicle, and below the seminal vesicle is a prostate, and then bulbourethra is there, and it goes into the bulbourethral gland, and then it in the whatever the secretion, whatever the glands, whatever they secrete, I mean, it goes to the urethra, and then the it comes out with the sperm and the whatever the comes out as a semen. Okay, now I will share the next. All right, now we will see one by one. So the paired testis is a primary sex organ that produces sperm and testosterone. Testosterone. So the testes are formed within abdomen. Soon after the birth or at the 8th month of the pregnancy, they descend into the scrotal sac. Actually, what happens is when they descend into the scrotal sac, this is at least 2.5 degrees Celsius less than the body temperature. So it helps in the proper functioning of the testis and for spermatogenesis. So it stays in the sac. So each testis is oval in shape, length 4 to 5 centimeter, width 2 to 3 centimeter. Each testis has about 250 testicular lobules. So each lobule contained 1 to 3 coiled seminiferous tubule. We saw the tubules, right? Here, see. Seminiferous tubules here in this. Okay. And the seminiferous tubule is lined internally with spermatogenesis cells, the spermatogonia or male germ cells, and Sertoli cells. Sertoli cells is basically supporting cells. I mean, it gives a shape and it gives nourishment for developing spermatogonia. So these are the Sertoli cells, and primary spermatocyte is this one. These are the secondary one, and this is the spermatozoa. And now the regions outside the seminiferous tubules, interstitial spaces contain what? It has small blood vessels, and then the interstitial cell or the Leydig cell. Leydig cell basically secrete testicular hormones like androgen. So, and immunologically competent cells are also there. So, three types of things are there in the seminiferous tubules, interstitial space. So, what are they? Blood vessels are there, and then Leydig cells are there, and then some other competent cells are also there. Now, Leydig cells is very important because they secrete the testicular hormone, that is androgen. Now, the accessory duct. What do they? Where are they, and what do they do? So, reti testis, that is the irregular cavities, and vasa efferentia are the series of fine tubules. Epididymis is the stores sperms and temporarily. I mean, the stores go there temporarily and then it is released. And then comes the vas efferens. I mean, it travels through the vas efferens. So, vas deferens joins with the duct of seminal vesicle to form common ejaculatory duct. Okay. So, see, this is the conduction of sperm through accessory duct is like this. See, first seminiferous tubules and then reti testis. And then it goes to vasa efferentia and then epididymis. It's stored there temporarily. And then from there it goes to vas deferens and then common ejaculatory duct and then urethra meatus. The urethra receives a duct of prostate and Cowper's glands and passes through the penis. So they include what? A pair of seminal vesicle, a prostate gland, and then a pair of Cowper's bulbourethral gland. So their collective secretion is called the seminal plasma. So this is rich in fructose, calcium, and enzymes. So the seminal plasma plus sperms is a semen. So function of the seminal plasma, what is the function? Why it is there? Helps for transporting sperms, supplying nutrients to sperms, providing alkalinity to counterpart acidity of uterus. Secretion of Cowper's glands lubricate the penis. So secretion of epididymis, vas efferens, seminal vesicle and prostate help for maturation and motility of sperm. All right, so this is about the male reproductive system. With this, we come to the end of the class. The rest of the unit we will finish in the next week, and then we will do the NEET questions connected to this unit. Okay, children?
Thank you, ma'am. Thank you, sir.